Hi everyone, welcome to Swati's Bioscience Hub. So till now we have been discussing about the cellular respiration and its stasis. And we have discussed that cellular respiration involves four main stages that starts with glycolysis, pyruvate dehydrogenase complex, Krebs cycle and lastly electron transport chain. Okay, so cellular respiration is required to convert the macromolecules which we get after the digestion of our food, carbohydrates, lipids and proteins, to use it for the energy synthesis or ATP synthesis. Okay, and in the last video we have discussed about the first step of cellular respiration, that is glycolysis. Okay, and we have discussed in glycolysis glucose, a 6-carbon compound, it follows 10 successive enzymatic reactions to produce pyruvate, a 3-carbon containing compound. Glycolysis is a carbohydrate metabolism pathway, and in glycolysis glucose follows 10 successive enzymatic reactions to produce pyruvate. Okay, and this glycolysis is always seen in every kind of organism. Okay, whether it is eukaryotes and prokaryotes, so it is always present in all organisms and in all conditions, aerobic and anaerobic, in the presence of oxygen and in the absence of oxygen. Glycolysis is present in every condition and in all the organisms. But then this pyruvate, so it produces glucose, it converts the glucose to pyruvate in glycolysis process. But after that, this pyruvate behaves and produces different products depending on the requirement of our body and the presence of oxygen. Okay, so this pyruvate, it produces different products. So first we will see when there is no oxygen, that means in anaerobic condition, no oxygen molecule. Okay, in the absence of oxygen, this pyruvate converts to lactic acid or lactate. Okay, it produces lactate or lactic acid. As example, when you do a lot of physical activity or exercises, our muscle cells produces lactate from pyruvate, because that time our muscle cells lack oxygens. Okay, and when there is a little bit of oxygen, that means hypoxic condition. Very small oxygen is present, hypoxic condition, that is almost anaerobic condition, and this condition is very rare. At that time, pyruvate produce to ethanol and carbon dioxide. Okay, so in anaerobic condition it produce lactate, and in aerobic condition it produce ethanol and carbon dioxide. In the presence of oxygen, in the aerobic condition, when the oxygen is present, aerobic condition, it produces acetyl coenzyme A. It produces acetyl coenzyme A. Okay, this acetyl coenzyme A that directly enters to the third part of the cellular respiration, that is TCA or citric acid cycle or Krebs cycle. Okay, this acetyl coenzyme A can further carry out the cellular respiration process. Okay, the aerobic cellular respiration process. So now we see in 3 different conditions pyruvate produces different products. In anaerobic condition it produces lactate. In aerobic condition it produces acetyl coenzyme A, and in the hypoxic condition it produces ethanol and carbon dioxide. And here this condition is very rare. Okay, this is very rarely produced. Now the conversion of pyruvate to acetyl coenzyme A, this conversion includes multi-enzyme subunits. Okay, this conversion includes multi-enzyme subunits that is known as pyruvate dehydrogenase complex. Okay, the conversion from pyruvate to acetyl coenzyme. It includes multi-enzyme subunits those are E1, E2 and E3. Okay, and this complex is known as pyruvate dehydrogenase complex. So there are three enzymatic subunits at present. E1 is pyruvate decarboxylase. It is pyruvate decarboxylase. E2 is dihydrolipoamide transacetylase. It is dihydrolipoamide transacetylase, and E3 is again dihydrolipoamide dehydrogenase. Okay, so these are the three enzyme subunits which performs this pyruvate dehydrogenase complex, which altogether performs the conversion of pyruvate to acetyl coenzyme. As the name suggests, each of three enzymes, they used their associated functions also same. Okay, like as their name suggests, enzyme 1 responsible for the decarboxylation process, enzyme 2 responsible for the transacetylase process, and E3 for the dehydrogenase process. So here what happens, this E1 is linked with the decarboxylation of pyruvate. Okay, E1 enzyme is linked with the decarboxylation of pyruvate. It releases the carbon dioxide molecule from pyruvate. Okay, so if we'll see the overall reaction, the basic reaction of pyruvate dehydrogenase complex, so that is, the substrates are pyruvate plus coenzyme A group, and it produces acetyl coenzyme A plus carbon dioxide plus NADH plus H plus ion. Okay, so this is the overall reaction of pyruvate dehydrogenase complex. So this E1, the pyruvate decarboxylase, as the name suggests, it is linked with the decarboxylation process of pyruvate. So here what it does, from pyruvate, in the help of E1, it releases carbon dioxide molecule. It decarboxylates the pyruvate, thus modifies it. When it will decarboxylate, it will remove one carbon group from it, it will modify the pyruvate and it releases the carbon dioxide. At the same time, next, the E2 enzyme, it will transfer the acetyl group to the coenzyme A group. Okay, it will transfer the acetyl group to the coenzyme A group, and here it will produce the acetyl coenzyme A, the final product of the pyruvate dehydrogenase complex, which will enter into the Krebs cycle. Okay, so now the question is, this pyruvate dehydrogenase complex are taking substrate this time, and the product should be acetyl coenzyme A. We are taking E1 and E2 enzyme units. So when we are taking E1 and E2 and we are getting acetyl coenzyme A already in the second step, then why do we need this E3 enzymes? Okay, the E3 enzymes. Here what happens, the question is, why do we need the E3 enzymes or E3 subunit for the process, when we have already getting the result from E1, from the second step of the reactions with the help of E1 enzyme and E2 enzyme. So question is, why we need E3? Actually, E3 helps in reducing the NAD plus to NADH. Okay, E3 helps in reducing the NAD plus to NADH and FAD to FADH. Okay, and this NADH enters into the electron transport chain and it couples with the ATP synthesis to produce ATP and energy. It produces ATP and energy in the electron transport chain. Okay, that's way, and somehow E3 in this, likewise this E3 re-establishes the whole process. It produces NADH. NADH enters into the electron transport chain and there it produces ATP, and E3 helps in re-establishing the whole complexes. Okay, the whole complexes. Now we'll see the mechanism of the pyruvate dehydrogenase complex, the brief mechanism of it. So, to generate acetyl coenzyme A, the pyruvate dehydrogenase complex first convert the pyruvate dehydrogenase complex first convert the pyruvate to an immobilized active acetaldehyde. Okay, it converts the pyruvate to an immobilized active acetaldehyde. That means it's not stable. It's not a very stable product. So then what happens? This and with the help of E1 enzyme. Okay, first is produce a immobilized active acetaldehyde with the help of E1 enzyme. Okay, and this E1 enzyme has a cofactor with it. E1 has a cofactor, that is thiamine pyrophosphate. That is known as thiamine pyrophosphate, denoted as TPP. Okay, so this thiamine pyrophosphate, it forms a strong nucleophilic carbon ion on this pyruvate. Okay, and this the cofactor of E1, it forms the thiamine pyrophosphate, that is a cofactor of E1 enzyme, and it forms a strong nucleophilic carbon ion. Then the carbon ion, which is formed by the TPP, it will perform a nucleophilic attack on the 2-carboxy carbon of pyruvate. Okay, so the thiamine pyrophosphate, it forms a nucleophilic carbon ion. Then this carbon ion will perform a nucleophilic attack on the 2-carboxy carbon of pyruvate, and it rearrange the structure of pyruvate, and here in this process it will remove one carbon dioxide molecule from pyruvate. Okay, when it will attack nucleophilically on the 2-carboxy carbon of pyruvate, it will release one carbon dioxide molecule, and then it will produce hydroxyethyl thiamine pyrophosphate. Okay, after release of carbon dioxide molecule, pyruvate rearranges its bonds, and it will produce hydroxyethyl thiamine pyrophosphate. Okay, E1 with the help of E1 enzyme, that is pyruvate decarboxylase. That means decarboxylation process takes place. Now, after the release of this carbon dioxide and formation of hydroxyethyl thiamine pyrophosphate, the next step, the carbon electron from this carbon electron from hydroxyethyl pyrophosphate, it will make a bond on the lipoic arm of the E2 enzyme. Okay, it will make a bond on the lipoic arm, first lipoic arm. Okay, on E2 subunit. Okay, E2 enzymatic subunit, that is dihydrolipoamide transacetylase. On that E2 subunit, this carbon electron from hydroxyethyl thymine pyrophosphate will go and form a bond with the lipoic arm. Okay, the first lipoic arm on E2. Then this group, this carbon electron group, again moves transfer from the first lipoic arm to the second lipoic arm. It transfers to the second lipoic arm on E2 subunit, and here it will transfer the acetyl group, that is carbon electron acetyl group, to the second lipoic arm, and here it produces the acetyl coenzyme A. Okay, at this point it will produces the acetyl coenzyme A. It will transfer the acetyl group to the first lipoic arm, then to the second lipoic arm, and here this acetyl group will link with the coenzyme A and will produce acetyl coenzyme A. So in the next step, with the help of E2 enzyme, that is dihydrolipoamide transacetylase, which is transferring the carbon ions or transferring the acetyl group from the hydroxyethyl thymine pyrophosphate to the first lipoic arm, then to the second lipoic arm, and it's producing acetyl coenzyme A. So now we can see that all the substrates are used. Okay, from pyruvate we got carbon dioxide. Okay, from pyruvate we got carbon dioxide, and then the pyruvate modified to hydroxyethyl. From hydroxyethyl we got acetyl, and acetyl along with coenzyme it produced acetyl coenzyme A. Now the final step is the production of NADH. Okay. So what happened here, the sulfide bond which are present in the E2 subunits, it reduces the sulfide bond on E2 unit, the sulfide bond on E2 subunit, that will be reduced, and along with it, it will reduce the FAD to FADH. It will reduce the FAD to FADH. Okay, with the help of E3 enzyme. Okay, it will reduce the FAD to FADH with the help of E3 subunits, that is dihydrolipoamide dehydrogenase. Okay, and this E3 subunits will transfer the electron from FAD to NAD plus. It will transfer the electron from FAD to NAD plus, and here it will convert that NAD to NADH. Okay, now this NADH will directly enter into the electron transport chain, and here it will produces energy or ATP, and the acetyl coenzyme A that we got from the second step with the help of dihydrolipoamide transacetylase, and this acetyl coenzyme A will directly enters into the Krebs cycle or TCA cycle or citric acid cycle. Okay, so this is the overall reaction and the brief mechanism of the pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex mostly consists of 3 multi-enzyme subunits, 3 enzyme subunits. One is E1, that is pyruvate decarboxylase. Second one is E2, that is dihydrolipoamide transacetylase, and the third one is E3, that is dihydrolipoamide dehydrogenase. And in this process, it first converts the pyruvate to the immobilized active acetaldehyde. Then this acetaldehyde, then the cofactor of E1 enzyme, that is thymine pyrophosphate, that will act as will form a strong nucleophilic carbon ion, and this carbon ion will perform a nucleophilic attack on the 2-carboxycarbon of pyruvate. At that point, this pyruvate will release the carbon dioxide molecule from it and will produce the hydroxyethyl thymine pyrophosphate. And all these things happens in the presence of this reaction is catalyzed by E1, pyruvate decarboxylase. Then this hydroxyethyl thymine pyrophosphate, it transfers one carbon electron, or that is acetyl group from it, and this acetyl group is transferred. It will form a bond with the first lipoic arm on the E2 subunit. Okay, and here it will transfer to the second lipoic arm. It simply transfer to the second lipoic arm on E2 subunit, and then it will produce acetyl coenzyme. And in the last part, with the help of E3 dihydrolipoamide dehydrogenase, this NAD plus will convert to NADH, and this NADH will enter to electron transfer chain. It is said for the electron transfer chain part, acetyl coenzyme will enter into the Krebs cycle, the third step of cellular respiration. So this is all about the pyruvate dehydrogenase complex. So if you have any doubt regarding today's discussion, please comment down below, and thank you so much for watching my videos, and please like, share and subscribe my channel. Thank you so much.